Okay, so we are now recording. Okay, so these learning objectives on the first slide are also found on Canvas in your resources section under each one of the chapters. These are what I tell students I use to design the exams, and these should guide your readings. And so as you read through these are pretty much the things that I expect you guys to be able to know and do as you're looking through each one of the sections. Okay, so I always like to start with kind of just basic true-false questions. These are some of the key points throughout the chapter that are things you should be able to pick up after you've gone through the lectures. So the first one here said that the ability of a microscope lens to differentiate between two separate objects is based on its magnification power. So in the little chat, guys, type a T or an F. Do you guys think that's a true statement or a false statement?
I don't have access to the chat. I don't know if I'm the only one.
I don't have it either. I'm trying to see if I can type.
Yeah, same.
I don't have it.
Yeah,
either.
if you move your mouse to the center of your screen, does a little bar pop up where it has your camera and your microphone?
Yeah.
And
it does, but it won't let
us there for the chat.
It says chat and channel meetings is only available to team members.
That's interesting. Don't worry about it for today, and I will figure out why you aren't a team member afterwards. So it's all right if you don't have the chat. You can ask me questions verbally if you need to. This is mainly just so I can get a little quick poll of the class itself. Okay, so I'm seeing lots of Fs pop up, which is true. So this is a false statement. And the reason this is a false statement is that when you look at the microscope, there's two key properties. There's magnification. There's also resolution. Magnification is just making things bigger. And so this is a false statement because it's not based on the magnification power. It's based on the resolution. That's an important distinction for you guys to know the difference of. And so to understand what the resolution of a microscope is, and looking at if two objects are close enough together, can you still determine that they're two separate objects? Okay, the second question has to do with parts of the microscope itself, which typically is easier when you guys have a face-to-face lab because it means you have used the microscope. And so I'm going to focus a little bit more on the microscope this semester than I typically do in lecture, just because you guys don't get to really use it in lab this semester. So this one says the condenser lens is an important component of a compound microscope because it narrows the beam of light to focus on the specimen. So what do you guys think, true or false? Good, I'm seeing some trues show up on the list there. That's a true statement. Compound microscopes technically have three lenses. They've got the eyepiece or the ocular lens. They've got the objective, which is the lens that you can change. And then they've got the condenser lens, which isn't actually involved with magnification, but is instead involved with getting that light focused on your specimen so you can get the best resolution that you can out of the microscope. Okay. If you're completing a differential stain, you must be using a positive stain and a negative stain. So that one is going to be a false statement. Differential stains are stains that allow you to tell the difference between different types of microbes, and they require you to use two stains. But the reason this is a false statement is they don't have to be a positive stain and a negative stain. And in fact, most of the time, they're actually two positive stains. And the difference between positives and negative stains, if you remember, positive stains have a positive charge to them, and so they're going to stain the cell itself. Negative stains have a negative charge to them. Most cell structures are also slightly negative, and so this is repelled by the cell and stains the background. And then the fourth true-false was that a wavelength of a beam of electrons is much smaller than the wavelength of a beam of light, allowing the electrons to have a much greater resolving power than light microscopes. And that's a true statement. So the resolution of a light microscope is based on the wavelength of light divided by 2 times the numerical aperture, which is just a property of the lens. And so the smaller your wavelength of light, the smaller the overall resolution. And the lower the resolution, the better the resolution of the microscope. So if you have a resolution of, say, 0.1 nanometers, that's telling you that those objects could be 0.1 nanometers apart from each other and you could still see them. As opposed to if you had a resolution of 500 nanometers, those objects, if they were closer than that, you wouldn't be able to see them anymore. So the lower the resolution number, the better the resolution of the microscope itself. Okay, so we're going to move on. We're going to kind of walk through the main topics of this chapter. The first was microbial size. And so this is just kind of a fun way to look at microbial sizes. I don't need you guys to memorize exact numbers for anything in this class. I just want you to have a general sense of what types of organisms are going to be in what size range. The other thing I'm going to be doing throughout the semester is I'm going to try to be using coronavirus where I can as examples, mainly so that you guys can learn a little bit about the ongoing pandemic while also learning about classes stuff without turning it into an entire class on COVID-19. And so that's why you can see coronavirus is on the list here. And so we have a size scale ranging from 10 nanometers all the way up to 1 centimeter. One of the tricky parts of micro is that most of the microbiology we're going to be working with is going to be down in this general range, which are much smaller sizes and much smaller units than most students are used to working with or really picturing things. So if you 1 millimeter, so if you're looking at a ruler, and we've got here, here's your 1 centimeter mark. So the metric ruler, because we're in science, so we do everything in metric. And then this is broken down into 10 pieces. Each one of these is 1 millimeter, okay, 1/10 of a centimeter. What you want to then picture, if on that ruler you picture that one little 1 millimeter line, and then break it down into one, then, and if you take one of those little lines and break it down to another 1,000 lines, then you would be at 1 nanometer. And so very, very tiny things that we're talking about. If we put these organisms, coronaviruses are about 120 nanometers, so they would fall kind of right around here. And that's to help remind you that your viruses are all typically going to be under about 500 nanometers. Okay, so anytime you're down in this range, we're going to be looking at virus cells. Okay, we'll be looking at virals. Let's see. Try to figure out why I'm breaking up for you guys. All right, I changed my microphone setting, so we'll see if that helps at all. Keep putting comments up there if I'm still breaking out pretty good. Okay, good. That's a start. All right, so what I had been saying was that viruses run into this about 500 nanometers or smaller size. E. coli, which is a prokaryotic bacteria, is about 2 micrometers. So E. coli would fall in here. And Methanobrevibacter is a prokaryotic archaea. They're both prokaryotic cells, and so they're both falling in to right around this range. Your Methanobacter are about 3 micrometers. Okay, so remember, this is your prokaryotic cell range. You've got both bacteria and archaea, 2 separate domains. Okay, just because they're single cell prokaryotes, they're actually very different from each other, but size-wise, they fall in about the same area. Next on our list were diatoms. Okay, diatoms are eukaryotic algae, and they are up at about 500 micrometers. Whereas your yeast, which are a eukaryotic fungi, are at about 10 micrometers. And so that just shows you the breadth of the eukaryotic microbes. Okay? They're typically somewhere between 10 micrometers and somewhere under 1 millimeter. And so all of these ranges are all microscopic. The naked eye can comfortably see slightly smaller than 1 millimeter. So anything lower than that is what we call microorganisms and what we need a microscope to view. So you guys should have a general sense of what the size ranges are for the various classifications of microbes. Okay, before we move on to microscopy, does anyone have questions about microbial size or anything that we've talked about?
I have one question in regards to the resolution equation.
Yes.
Could you repeat, you said the smaller the wavelength, that is the lower the resolution and then therefore the clearer the object?
Yes.
Okay.
Yeah.
The
And
lower
so
resolution is good.
Yeah, so the lower resolution, the better the microscope.
Perfect. Perfect. Thank you.
No problem. Other questions? Okay, so let's work on to actually using one of those microscopes. So this is kind of your standard compound microscope that just about everyone has at least seen at some point. As we look at the overall components of it, there's really three things that you do with the microscope. You need to be able to magnify the image so that you can see it. You have to be able to change the depth of field so you can put it into focus. And you have to be able to have the light shining on the object so you can actually view it. And so the parts of this that are involved in image magnification, going to be your ocular lens and your objective lenses. These objective lenses are different powers. And so you typically have, this is a 10x lens, this is a 40x, and this big one here is a 100x objective. Whenever you're doing magnification, the total magnification of the microscope is whatever objective you have set multiplied by your ocular lens, and the eyepieces are pretty much always 10x. And so if you have, say, a 10x objective set above your slide, you're viewing the image 100 times larger, so you're 10 times 10. If you've got the 40x objective, you're looking at something 400 times bigger than it actually is. And if you've got the big 100x objective, you're looking at something 1,000 times bigger than it actually is. If you were in the lab using the microscope, this is the objective you need if you're going to be viewing prokaryotes. Things that are down in that 1, 2, 3 micrometer size, you have to have the 100x objective to actually be able to magnify them large enough to see. The eukaryotes, we tend to use at the 40x, and the 10x objective is really just to use to find your specimen, and then you kind of magnify in from there. In order to make sure your specimen is in view, it has to fit right along the focal point of the depth of field. So what's going to be coming out of this light down here? So here's your light source coming up. You're going to have a beam of light that's coming through there. And what happens is that beam of light, so here's our lens, that beam of light passes through, it gets refracted down, and wherever those points meet is where you're going to be in focus. So these knobs here on the side will actually move the stage up and down. And what you're trying to do is move it so that wherever that focal point is, the stage is right there, and then you can actually see whatever your specimen is that you're looking at. And we already talked about the condenser lens. The condenser lens is here underneath the light microscope, so that as the light comes up, we'll actually put a different color so you can see it on the microscope. So as the light comes up, it's going to get folded over and refracted so that you've got a nice pinpoint of light coming up into the actual objective. And so that's the basic components of how a compound light microscope works. In the lab, you'd be getting a chance to work on those. Unfortunately, you're doing it online, so you'll have simulation. You'll be able to work with the microscope, but I wanted you to have a general sense of what you were actually looking at when you were going to be doing those things. Okay, so now we're going to move into the meat of this chapter. This one is typically where students have questions. So in the lecture slides, I walked through how you calculate magnification. You have a whole worksheet on it to practice and a little quiz. And so I want to make sure that everybody is comfortable going through these. So I'll walk through this one instead of asking you guys what you think the answer is, but make sure as I'm going that these are the types of things that you're practicing with the worksheet and that you can do and understand on your own. Okay, so if we're going to calculate magnification, the biggest thing when you're looking at anything that comes out of a microscope is to remember that this is an image. Okay? Anything you can see on this picture is an image. It's blown up and magnified big enough so that you can see it. And the key to know how much bigger this is than the actual organism that you're looking at is what's called the scale bar. And this used to be a lot easier to understand when people used maps for real, because the scale bar on these images is very similar to the distance bar on a map that will tell you, say, 1 inch on the map is equivalent to 1 mile in real life. This is the same thing. It's telling you that this scale bar measured 5 millimeters when we put a ruler here and measured it. So that 5 millimeters in this image is equivalent to 50 nanometers in real life. Mm-hmm. So our magnification equation is going to be that the magnification is just the image size. So what size is the image you're looking at, divided by the actual size of the organism. And the reason we care about the scale bar is because right now, looking at this image, it's the only thing we know of that has numbers for both of these. So we know the image size was 5 millimeters because that's what we measured with a ruler. Remember, if you can see it and you can measure it, it's an image. And the actual size is what's written on the image itself. So in real life, that little bar there is only 50 nanometers in size. And so this is a pretty straightforward equation. The most complicated part of this is remembering what is your image size and what is your actual size, and then it's being able to do your unit conversions. There's a whole video on Canvas that's how to do unit conversions. So if you're not comfortable converting metric units, take a look at that. You can also sign up for online office hours with me if you want me to walk through how to do those unit conversions. I do them in my head. And the way I do them in my head is that I always go, so if you have a larger unit going towards a smaller unit, the decimal point is going to be moving in the direction of this arrow. So that means the decimal point would be going to the right. If you're going from a smaller unit to a larger unit, it means the decimal point is going to the left. And then the number of spots the decimal point moves is based on how many zeros, right, how many tens are in between the units. So a millimeter is 6 times bigger, or 6 units, zeros bigger than a nanometer. So if we were going to do that conversion, millimeters are larger than nanometers. So I'm going to the right. And I'm going 6 spots, 1, 2, 3, 4, 5, 6. And so 5 millimeters is the same thing as 5 million nanometers. I divide that by the 50 nanometers, and I'm going to get that the magnification of this image was 100,000 times. Okay. Notice your units cancel out. There's no real unit to magnification. It's just how many times bigger is something. And so we typically just put the X there as a times larger. Okay, so let me pause there. Does anyone have questions on how to use this equation? Okay, everybody seems to be okay. So now we want to think about what that number means. So based on that magnification, okay, of 100,000x, is that image taken with a light microscope or an electron microscope? So go ahead, type in the chat. What do you guys think? Is it a light microscope or is it an electron microscope? Good. I see a whole bunch of electrons showing up. So this was taken with an electron microscope. And the big key giveaway there is the magnification. And so this image was magnified 100,000 times. Remember, the maximum magnification of a light microscope is 1000x, 1500x if you get a really, really good light microscope, but there's no way a light microscope is magnifying 100,000 times. And so that's how we knew, is that our magnification range was outside the range of a light microscope. I'm always telling students, I tend to test on how you can use and apply the information. This is the type of questions that I mean. I'm not going to expect you to spit back at me what the total magnification for a light microscope compared to an electron microscope is, but you should know that information to be able to use it and answer questions like this. The next question we wanted to know, based on the image itself, what specific classification of microscope was it? So we talked about 4 different types of light microscopes. We've already decided this is an electron microscope, which means this is either going to be a scanning electron microscope picture or a transmission electron microscope picture. And if we go back and we look at the picture itself, this is not the surface of something. This is the internal structures that we're looking at. And so that tells us that this is going to be a TEM picture, because this looks at the internal structures, whereas SEM is really just looking at the surface of things. The image itself, this is actually a Golgi apparatus. So this is the inside of a eukaryotic cell, and these are all those stacked membranes that make up the Golgi. Okay. The last part of this problem then wants us to say, okay, typically we want to know how magnified an image is, not because we care about the magnification, but because we want to know how big the specimen is that we're looking at. And so now we're going to come back to that same equation, but we're going to use it differently because now we know the magnification. So remember, we were magnification equals image size over actual size. But now we know that this is 100,000 times magnification. And what we're trying to find out is how large is the actual Golgi apparatus? So what is the actual size? So we're going to be solving for A. And so we need to fill out I. We have to have a value there. And our problem tells us the red bar, which we can see here, measures 20 centimeters with a ruler. Remember, anything you're measuring with a ruler is an image. And so this value is 20 centimeters. We're going to rearrange this to get A by itself. So multiply both sides by A and then divide by your magnification. So we get 20 centimeters divided by 100,000x. So the actual size of that Golgi apparatus is going to be 0.0002 centimeters. That's a really tiny number. It's because in microbiology, centimeters are way too big for most of the stuff we work with. If we want to change that into a more normal looking number, that would be the same as saying this is 2 micrometers in size. But unless I tell you in a problem that you have to do a conversion, hey, either one of these answers is correct. The biggest thing to watch for is in multiple choice questions. Say you calculated this, but the answer in the multiple choice questions was 2 micrometers. You're going to have to make sure you're comfortable changing back and forth between these. So if this was an option in your multiple choice, make sure to do the conversions as you're looking for what the right answer would be. Okay, I'm going to pause there again. Questions on calculating magnification or object sizes from a microscope picture?
I have a question.
Yeah.
I guess it's not about like the math or anything. I'm confused on how like in the previous picture, we like had the actual size and now we don't, and we're solving for it.
Yep.
Does that make sense? I guess I just don't understand the difference.
Okay, so here's the key. The only way we had an actual size here is because of the scale bar.
Okay.
And so this is the importance of the scale bar. It's the whole purpose of having a scale bar on micrographs. So images that come out of a microscope, when they're published, should always have a scale bar on them. The reason for that is this is a property of the microscope itself. So in the microscope, there's actually a little bar in there. And on the image, you'll notice that that little bar has this 50 nanometers written underneath it. That's the actual size of the bar. And so in the microscope, there's a little tiny bar that's 50 nanometers in size that you would never be able to see with the naked eye. But when you look through the microscope, now you can see that bar. And because it's now big enough, it's been magnified, we can measure that bar with the ruler. That's the only reason we knew the actual size. If you looked at this image and it didn't have a size bar, you would have no actual sizes on that image at all, because you have no idea what size the cells are that you're looking at. You just know you're looking at cells. The only way you get an actual size is because of that scale bar. And so the first step is to calculate the magnification from the actual size of the scale bar. And then the second step is to use that magnification to calculate the size of what you actually care about, which is the specimen.
Okay.
Does that help?
Yeah, so the second one, it's the actual size of exactly what you just said, the specimen, whereas the first one, you're just using it to understand how much it's magnified.
Right. And sometimes a problem will tell you the size of the magnification. So I could have told you in this problem that this image was magnified 100,000 times and you've measured the size of the Golgi and it's 20 centimeters with a ruler. If that's the case, then you wouldn't need to use the scale bar, because I would have already told you the magnification. But if you just have an image and it doesn't have a magnification associated with it, you have to use the scale bar to calculate the magnification, and then you can use the magnification to answer the rest of the questions.
Gotcha. Thank you.
Yep.
I have a question.
Yep.
Will you ever want us to get a ruler ourselves? So, for example, if you gave us the scale bar, would you ever want us to get would you want us to measure a ruler of the scale bar to get the numerator portion of that fraction? Or
That's a good question. So in real life, that's what you would do. In class, I typically will just tell you what it measures, because it means you will all come up with the same answer. So if I have you guys measure it, some person might measure it 5.1 millimeters and some might measure it 5.5. Those are all close enough to be real answers, but they wouldn't show up on a multiple choice. So I'll always tell you the size, as opposed to having to have you guys try to measure stuff on your computer screen and things like that.
Thanks.
Okay. I also have a question that was up here about what determines the size of the scale bar. That actually is completely set based on what your image is and what magnification you're set at. So a lot of times this is done with a computer, where you can, especially in electron microscope pictures, you can determine how big you want that size bar to be. So you tell it, put a bar that's 100 nanometers on here, and it will make the bar based on what the actual image is that you're looking at. In a light microscope, it tends to be just a set property of the microscope itself, and so that's based on the manufacturer usually. And so those are things that are just part of the microscope and the imaging software that they come with.
And then you mentioned that we would have a little quiz on this. Is that just on Canvas?
Yes. Yeah. So on Canvas, it's labeled the microscope practice quiz. There's in OneNote, there's a worksheet that actually has you do just this. So it has you walk through practicing these problems. And then the quiz is based on the answers that you get while you're practicing those problems. Any of those types of quizzes, the day after the quiz closes, the answer key to those worksheets shows up on Canvas. And so after you've done the problems and you've done the quiz, you can always the next day look at the answer key to check for things that you did wrong. Okay, any other questions? Okay, so I'm just going to briefly go through the rest of these. A lot of these are typically things that students understand pretty well. They're more just kind of knowing the difference between things. Okay, so if you do a staining procedure using a single stain that turns your background blue, what type of stain is this? So is it a differential stain using a positive stain, a differential stain using a negative, a simple stain with a positive stain, or a simple stain with a negative stain? So the answer to this question is going to be D. We used a single stain, which tells us that this was a simple stain. Okay, simple staining procedures use one stain. Differential staining procedures use at least two different stains so you can tell things apart from each other. And then positive stains, stain the cell, negative stains stain the background. And so we've turned the background blue, so that tells us the answer to this one is D. Again, this is how I like to ask these types of questions. I didn't just say, give me the definition of a negative stain. Okay? I'm not looking for you guys to memorize things and spit them back. I'm looking to see, okay, you've actually done something and this was the result. What did you actually do? So you have to understand what simple stains and differential stains are, what positive and negative is to be able to answer this question. But it's a little bit more than just memorize a definition and spit it back to me. Okay, we'll take a couple minutes just to look at different types of light microscopes. There's 4 main types of light microscopes that we're going to be looking at. This one wants to know which one of these images is showing a bright field microscope image. It's going to be this middle guy here. So the big key with bright field is we have stained cells in a light background. Okay, the light is coming through all through the background, and most bright field microscopy requires staining cells for contrast so we can see these stains. The key with bright field to remember is that any stains that you've done for bright field microscopy mean the cells have to be dead. Okay, when you fix the cells so that you can stain them, which typically means you've put them through the Bunsen burner flame, means those cells are no longer alive. These other 2 images, this is fluorescent microscopy over here. We can tell that because we've got this black background. There's no light coming through there, but we have this nice fluorescence. So this light being emitted from the cells themselves. And this image over here is your DIC. And we can tell that because we get a bit of a 3D effect. We can see some nice contrast in them. And the key with DIC or phase contrast is those cells can still be alive. And so again, with these, when you're looking at kind of studying and answering questions, the types of questions I like to ask about the microscopes are more like these. Okay. Why might you want to use one microscope over another? In order to do that, you have to be able to say which properties of a microscope they have, but I don't care if you guys know the inner workings of a microscope. Unless you guys are going to work for Zeiss and build these things, it doesn't matter to me if you understand what's happening with the lenses and the light. I want you to know why you might want to use one over another. Okay, so if we wanted to follow the localization and movement, so where and how is it moving of a specific protein inside a cell, what type of microscope would we want to do? And I'm going to walk you through the logic of how to answer these types of questions. So things to think about. We're looking for localization and movement. And so the first step is if anything's moving through cells, those cells have to be alive. Okay, and so that eliminates something like bright field, where the cells are going to be dead. And we want to look at a specific protein. Okay? Proteins are really tiny, even smaller than viruses. And so the only way to really see a protein in a light microscope would be if we labeled it with a fluorescent dye and looked at it under the microscope. So we would want to use a fluorescent microscope for this first one. If we wanted to observe internal structures of a virus, okay, we want to think about, well, what type of microscope would we use to look at viruses? That's going to have to be an electron microscope because light microscopes don't magnify enough to actually view a virus. And so if it's a or if it's an electron microscope, then we say, okay, we want to look at the internal structures. That's going to be a TEM. So those transmission electron microscopes will let you look at those internal structures. Swimming motion of a spirochete, swimming motion, again, alive. Okay. Spirochetes, we mentioned, is the cells that are most often used in dark field microscopy. And the other option sometimes students think about on this one is DIC or phase contrast, because the cells are also alive in those. The key with those is they're typically used for eukaryotic cells. You typically aren't getting the kind of resolution down at the bacterial level. And so this would really be a dark field scope. But the next one, looking at motility of eukaryotic microbes in pond water, that's a good phase contrast or DIC scope. And then we're looking to see, okay, trying to identify an unknown bacterium and if it contains a capsule structure. So we're doing a differential stain. Okay. Differential stains, that's going to be a sign and a big key that these are going to be bright field. And so I'm looking at the questions. Yes, so bright field is the only one that you're pretty much guaranteed to be dead in. Fluorescent microscopy, it's 50-50. So fluorescent microscopy, they can be alive or dead. And then DIC and your dark field, those are going to be living cells. And actually, I take that back. Bright field is the only microscopy for light microscopes. All electron microscopy are all dead. So you can't bombard something with a whole beam of electrons and expect that something to be alive. And so in electron microscopy, everything is dead, but in light microscopy, bright field is the only one where you're pretty much guaranteed that your specimens are dead. Other questions about the types of microscopes and kind of how you think about when you might use one over another before we move on to chapter 5? Okay, so again, here are the learning objectives for chapter 5. As you guys are thinking about kind of importance of material, one thing I want you to look at is when you go through my lectures, look at the learning objectives and think about how much time gets spent on different parts of things. The big key for chapter 5 is gram positive and gram negative. I want you to understand the general function of a lot of those other structures, but I really need you to know the kind of differences between gram positive cells and gram negative cells. So again, the things that I give you guys worksheet assignments for that have those worksheet quizzes, those are being given not as busy work, but because those are the key things that you can guarantee will be on the exam. A lot of this other stuff, parts of them will be on the exam, the functions of all those other structures. But as you're looking at where are you spending your time, anything that's been put into those worksheets, I mean, those are the really important parts of the chapters. And so this first sheet is really asking you to say and identify what are properties of gram positive cells and what are properties of gram negative cells. I'm actually gonna insert a new slide in here for a minute. Okay, so if you remember, okay, your cells are all going to have a plasma membrane. Okay, so this is the cell membrane. For gram-positive cells and gram-negative cells. All of your bacterial cell walls have what's called peptidoglycan in them. Peptidoglycan is this big complex molecular structure that's made up of 2 repeating sugar molecules, and they're held together by cross-links with proteins. But the difference between the 2 cell types is those layers of peptidoglycan in a gram-positive cell, we'll put our peptidoglycan in blue, is we get lots and lots of layers. So these are all big mesh networks of peptidoglycan, whereas in the gram-negative, you really only get 1 or 2 of those layers. So we get a very thick layer here, very thin layer here, but they're both peptidoglycan. But then in gram-negative cells, we have an outer membrane that's a lipid bilayer with a surface of carbohydrates. And those carbohydrates are called lipopolysaccharides. It does not say saccharide. So lipo, because they're part of the cell membrane, so they have a lipid component because they have this big sugar. Some of the other key differences is that all throughout here you have a periplasm, and this periplasm in gram-negative cells is actually used, whereas in your gram-positive, you just have a thin periplasm. So all through here you can have molecules, a very thin periplasm in your gram-positive cells. The only other structure that we mentioned in the videos is teichoic acid. which can either be wall-teichoic acid that's only in the peptidoglycan, or lipoic acid. So those are the basics of your cell membrane and your cell walls as you're looking at them. And so when we come up here and we look at the different components, we should be able to go through these pretty easily now. So a thick layer of peptidoglycan, gram-positive cells. The outer membrane of lipopolysaccharide, gram-negative cells. The cell wall functions to counteract osmotic pressure. That's both of them. That's the general function of a cell wall overall, is to provide protection to the cell. Colored purple after gram staining. Okay, we'll talk about the gram stain in a minute, but purple and positive both start with P. Okay, so your gram-purple cells or gram-positive cells are purple. Okay, a thin peptidoglycan layer we said was gram-negative. Contains peptidoglycan, which are made up of your acetylglucosamine and acetylmuramic acid sugars. That was both. So even one's thicker than the other, they both have that peptidoglycan structure. Teichoic acids were only in gram-positive. Now here's one we haven't necessarily talked about. Contains the transpeptidase enzymes to cross-link the NAM, so your N-acetylmuramic acid subunits. Remember we mentioned those sugars are all cross-linked together with your amino acids, and this is the enzyme that does that. Because peptidoglycan is found in both, they both have to be able to make those cross-links. So that enzyme is found in both of them. And I put this on here because that enzyme becomes important. That's actually the target of penicillin. So the reason penicillin kills bacteria is it stops that enzyme from working so they can't make their cell walls. Okay, colored red, those are your gram-negative. And remember, we say red and not pink, because if you say pink, that whole purple positive thing doesn't work anymore. So gram-negative cells are red. And oligosaccharides, okay, so saccharide should give you a key here. These are the sugars that are part of the lipopolysaccharide. So that would be an N. Okay, before we stop for questions, I'm going to actually take a look at the gram stain itself, and then we'll see if you guys have any questions on gram-positive and gram-negative overall. Okay, this is one of the ways I like to ask these types of questions, in that these are going to ask you what happens if you did something wrong. Okay, again, this is one of those use and apply type concepts where it's not just what are the 4 steps of the gram stain. It's do you understand those steps enough to be able to say what would happen if you didn't do one. So before we read through the actual things that it's looking at, remember the gram stain itself uses crystal violet as the first stain, which stains everything purple. You then use iodine, which is a mordant. It just makes sure the stain sticks to everything. Then you use ethanol. Ethanol is what we call the differential step because it's going to remove the dye, but only from gram-negative cells. So that's how we're now going to be able to tell the difference. That's why it's called the differential step. And then 4 is the counter stain, which in the gram stain is safranin, which is red. And that's just so that we can now re-dye the cells that we removed the stain from. Okay, so these are kind of our 4 steps. So let's see if this first question wants to know if we began the staining process with the iodine step, and so we forgot the crystal violet. So if we started here instead, I'm going to have you guys actually type this one in. When you got to the end of this, what color do you think your gram-positive bacteria would be? Remember, they were supposed to be, so after we add the safranin, our gram-positive would have still been purple, and the safranin would have dyed our gram-negative red. Okay, that's what's supposed to happen. If we started at iodine, what color do you think the gram-positive bacteria would be? Purple or red? Or clear? Okay, so I'm getting some mixes of answers. Got some reds, some clears, and purples. Okay, which means we're a little confused on this one. So let's take a look. Okay, so if we start here, we add iodine to the cells. That doesn't do anything because there's no dye there to make stick. So the cells are all still clear. Now we add the ethanol. The purpose of that ethanol was supposed to be to remove the dye from the gram-negative cells, but there's no dye there to begin with, so everything is still clear. Now we get to our safranin, our counter stain, And this is just a simple positive stain. And so when we add it to those cells, because everything was clear to start with, everything is going to turn red at the end. So the reason gram-positive cells stay purple normally is mainly just because they're already dyed. So the purple dye is already stuck to them, and there's no room for the red dye to get in there. And even if one or two red dye molecules did, purple is darker than red, so you don't really see the red anyway. Okay, so in this example, both of those cell types would end up red, and when you looked in the microscope, you wouldn't actually be able to tell one cell from another. And so you would have failed your gram stain. You wouldn't have been able to tell if you were looking at gram-positive cells or gram-negative cells, because in this instance, they all would have been red. All right, so now let's look at this one. Next one says we forgot to add the ethanol wash. So again, now this time we would have stained everything purple. We would have gotten that dye to stick with the iodine, but then we would have never removed the dye from the gram-negative cells because we would have skipped over the ethanol step and gone right to safranin. If everything is still purple when we add this dye, everything is still going to be purple when we look in the microscope. So just like if you do them normally and those gram-positive cells are purple, they don't stain red because the purple's already in the way. If you never removed the purple dye from your gram-negative cells, everything's going to be purple when you look in the microscope, even if you've added the safranin. And now this last one, if we forget to add the safranin dye at the end, well, our gram-positive are still going to be purple, but if we don't add the counter stain, we've removed the dye from the gram-negative cells, we never added another dye to change their color, and so they're going to be clear. Okay, so I'm going to pause there for a minute. Do you guys have any questions about gram-positive versus gram-negative, just for structures, for comparisons, or for the gram stain at all?
I have a question. Oh, sorry, go ahead.
Oh, well, I was just going to say, could you go back to the other side real quick? I just wanted to take a picture of it so that I could study it later.
Okay, yeah, also keep in mind, I'm recording this, so I'll put the recording up on Canvas so you guys can always listen to any of this again if you want to.
Oh, perfect. Thank you.
Yep.
Yeah, so I have a question. So are the stains, they're both positive stains?
Yes.
Oh, and then so the gram-negative and the negative and positive on grams don't refer to any charge. It's just...
No, they don't. And that's a good question. Scientists don't always name things in the most easy way to understand. And so the guy who developed this, his last name was actually Graham. And so he was actually looking for the positive color. And so the one that was positive that turned purple became positive. But yeah, those positive and negatives have nothing to do with charges whatsoever.
Great, thank you.
Yep. Any other questions?
I have a question.
Yes.
So from either the videos or the book, but it mentioned that basically we use the step 4 to stain the gram-negative, kind of only because after a while the purple will lose its stain and everything will go clear. Is that true? Because if at the end of it, if you have purple and then clear, you would know the difference, but you could identify the difference between the two. So why would we want to stain again if we can already identify the difference? Is that?
Yep. That's a good question. The biggest reason is, and this is again one of those things, actually I'll turn my camera on while I'm talking to you guys. One of the things of not being able to actually use the microscopes for lab is that you won't get to see this firsthand, but bacteria, even at the 100x objective, so at 1000x, are still really tiny in the light microscope. And so if they're clear, you pretty much can't see them. And so what's happening is you obtain one purple. If you remove the dye from the other one, they're really, really hard to see. And so the only purpose of the counter stain is to make sure you've got contrast between the light background and the cell itself, so you can actually tell they're there. Because a lot of times you might have what we call a pure culture. So you only have one species and you're just trying to figure out, is it gram-positive or gram-negative? If you did all this, but you didn't do the safranin and it was gram-negative, You might look in that microscope and just not see anything. And then you're not sure if you did your stain wrong or if there's cells there or not. And so realistically, that's the only purpose of the counter stain, is so that you can increase the contrast so you can see the cells better. The other key with the gram stain is conventionally it's purple and red because it's your crystal violet and your safranin. But there's a number of folks out there who are red-green colorblind. And if they are, they can't use the regular gram stain because they can't tell the difference between purple and red when they look in the microscope. So the actual dyes can be changed, and there is a color-blind version of the gram stain that they use different dyes, which means the cells change different colors. So the dyes themselves aren't nearly as important as the order of the steps and the fact that that ethanol is there as a differential step. That's really the key step of this. Other questions? Everybody good? Okay, so I'm just going to take just a couple quick minutes to show you how I tend to ask questions about the other structures of the cell, and then we'll open it up for just general questions if you guys have any.
Hey, sorry, I put one question in the chat. Sorry.
Oh, sorry, missed it. Okay, so I decided for the summer class, so the question she had in the chat, if you guys don't have it open, is do we need to know the difference in the detail of firmicutes and proteobacteria and some of the other classifications of how bacteria are classified? Typically, when I teach this class, you do. For this semester, I actually took that section out. And so we're actually going to skip. There's a whole chapter that goes into detail about some of those. Because this is online and in the summer, I care more about you understand why gram-positive and negative are there. Don't worry about the firmicutes and the proteobacteria. If I talk about those, I will give you the information that they are gram-positive or gram-negative so that you can answer the questions. Okay, so just briefly, let's take a look at just a couple of questions so you get a sense of how I ask about the functions of all those other organelles that the chapter talked about. Or not organelles, sorry, we're in prokaryotes, not eukaryotes, all those other structures that we talked about. Again, I don't like just spit back definitions. I'm not going to say, what is the function of a pili? Because one, your exams are open books, so you could just look that up and doesn't really tell me anything other than you can read. And so these are how I tend to like to ask these questions. So if you're studying a bacterial pathogen that you discover cannot be phagocytosed by cells of the immune system, based on this, what structure does the species likely contain? Okay, so this is how I tend to ask these. It means you have to understand what the functions of those structures are, what properties are they giving to the organism, but it's not just a memorize and spit back a definition. And so as we look at the answers, flagella, capsule, fimbriae, or pili, the easiest way to answer these types of questions, if you're not sure, is to look at each one of them and think about, okay, what was the general function of that structure, and would that possibly play a role? Okay. And so the answer to this one was the capsule. The main purpose of having a capsule is that it hides the cell a little bit, so it helps it prevent it being phagocytosed. It also helps it to not dehydrate. It helps it keep it a little bit hydrated. Okay. Another example of how I do these. So in order to colonize and invade the intestinal cells, foodborne pathogens must first adhere to the intestinal cell wall. So again, we want to pick out the key information. Okay, so they're colonizing and invading. They're adhering to the intestinal cell wall. What is a structure that they may use to do this? Okay, so again, our key here is that we're adhering. So what structures were involved with adherence? Okay, remember those were the pili and the fimbriae. As we look through our answers, there's our pili. That's what we're looking for in those. Okay, so that's how I ask these types of questions. You guys have the lecture videos, and so you can go through the general information of all of those. Just as one other kind of practice example for you guys, these are just not multiple choice versions, but ways that you can test yourself a little bit more to see if you can pull it out of your head. Bacterial species contains a gene that provides resistance to antibiotic penicillin. The gene is not part of the bacterial chromosome, and it's able to shared within the bacterial cells, leading to spread of resistance. So the key for this is the gene was not part of the bacterial chromosome. Okay, one of the key structures we talked about were extrachromosomal DNA pieces, and those extrachromosomal DNA pieces were called plasmids. Hey, they were the little bonus pieces of DNA that some bacteria have that can carry extra beneficial genes. Those are genes they don't need to be alive, but can give them beneficial properties depending on their environment. A bacterial species is able to perform photosynthesis because it contains the machinery localized to certain areas of the cell. Localized to certain areas of the cell, those are our inclusion bodies. And even more specifically, the specific inclusion body involved in photosynthesis were the thylakoids. We're placed on a petri dish with nutrients found in oil on one side, and the species can detect it and travel across the plate to get it. The key part of this is we're traveling. And what structure was involved in traveling for the microbes? Those were the flagella. I also like this because it points out, remember, bacteria aren't just kind of floating around willy-nilly or swimming all over the place without knowing where they're going. They actually can orient themselves and do chemotaxis. They can travel towards good things and away from bad things. They can orient themselves based on things like the magnetic pole for some of them. So they're not just these kind of little single cells that are at the whim of their environment. They do have control over what they're doing. And then this last one has a vegetative cell that died 75 years ago when the nutrients got scarce, but its genome is protected. And so what structure was it that was able to protect bacterial genomes and last for years and years and years? Those were the endospores. Okay, the last slide here, this isn't, again, remember, you can get this on Canvas. This isn't anything I'm going to go over, but this was just to help you guys study, because it allows you to tell the difference between bacteria, archaea, and eukaryotes. So kind of what were some of the size comparisons? What are some of the differences between their membrane lipids? This is just to help you guys study. So it's just there in case you guys want to use it at some point. Okay, so that's the end of these slides. I'm going to stop sharing my screen for a minute and see, do you guys have any questions for me about anything else? Either anything we've already talked about, anything from the 2 chapters, just anything about the class itself, because it's the first week and you're still figuring out what's happening.
A question that I have is, as we are studying and as we're listening to the lectures, I've just been taking a bunch of notes, but is there a study guide at all for the test that while we're listening to the lectures, we can review that?
Yeah, so one thing I always tell people that, so the learning objectives are your best study guide. They are found either in this set of PowerPoints or they've got their own page in the week's module. So if you look in the module for the week, there's a chapter 3 heading, it's got the objectives. Those are what I tell students to use as your main study guide. So I don't build kind of a outline study guide for you guys. As you're doing the readings and listening, look at the things that I expect you to be able to know and do in those objectives and use those to guide your readings. In the exam module, there's also a set of study questions that are just kind of practice questions that you can also use to help as a guide for what I think is important and not.
Okay, perfect. And then also it sounds like those practice questions that maybe, I think you said it were on the assignments, that would be a good thing to use to study as well.
Yes. And so I typically tell students I'm not a huge fan of busy work, especially in the summer. There's not a whole lot of time. All those worksheets are because they're covering key parts of the different chapters. And so after you've done them and the due date's passed, when the answer key shows up, that's a good way to use them to say, okay, the answer keys, I don't have just say like the number. I actually work through the math. So if you struggled on some of them, if you got some questions wrong on the quiz, you can pull up that answer key and use those to study. And then if you want practice, you can also go back to the blank version and kind of do them again if you need to.
Thank you.
Yep.
I have a quick question about each week.
Mm-hmm.
So you've posted on there all the lectures that are for the first week, I'm assuming, right?
Yep.
And then, because I had a class that she posted the entire semester, but she didn't always follow where she was or the guideline that she had originally thought to use, and so it got kind of hard to figure out where she was in the class. But if you're just doing it a week at a time, so it's easier to manage time and get the stuff learned.
So I'm mainly doing it a week at a time because next week's videos aren't made yet. They're about half made. So I'm pretty much going to be a couple days ahead of you guys. My goal is to always have your next week's stuff up by the end of this week. So all the videos and everything for next week will be up by tomorrow, so that as you start finishing up this week's assignments, you can begin starting next week's assignments, especially because those video lectures will be due next Tuesday as opposed to the Thursday, since we're now getting into the real weeks. And so, yeah, they'll get up. My goal is to get them up a little bit before the end of each week. We'll see if I get there, but they'll always be up for your next week by the Friday of the week before it.
That sounds fantastic. Thank you.
Yep. Okay, I have a question in the chat that says, how do we sign up for office hours? Let me actually show you guys, because that's probably going to be the easiest. Let me share my screen. I'll just share Google with you guys. Okay, so here's our Canvas page. What you want to go to is go to the actual calendar for Canvas. So over here on the side, you'll go to the Canvas calendar. In the calendar, make sure, so I've got Micro 2060 up. What you will see is, if you notice on there, there's all these little online office hours spots. When I highlight over them, they say unreserved. That's because no one signed up for them. What you guys can do is, when you're in the calendar, if you click on any of those, because you're not a professor and you're not editing them, it will give you the option to sign up. And so you can just come in. They're set for each week. So if we go down, next week's are already there also. And they're set in 15-minute increments. You're allowed to sign up for 2 of them. So if you just have a quick question, just go ahead and sign up for 1, for whichever one works for you. If you think something's going to be pretty complicated, go ahead and sign up for 2 back-to-back. You'll get your 30 minutes in. And then on Fridays, the case study office hours, this is times for your entire group to meet with me together. Only one of you from the group has to go ahead and register for those or sign up for those, and the whole group will be signed up. And so you can find them in the Canvas calendar. Just click on whatever times you want to. If someone has registered or signed up for them, they will actually say it when you hover over them. They're set so multiple students can sign up for the same one, but that way you'll know if someone has already signed up for that time or not. Hey, does that help?
Yes, I just put another question. So on mine, I'm not seeing like the office hours. I'm only seeing the assignments due on the days. I didn't know if there was a setting I needed to change or what.
Let me check.
I figured it out.
Has anyone else looked? Can anyone else see them on their calendar?
Yeah, I figured it out. If you go to appointments on the right-hand side of the screen above like all your different calendars, you can choose click on find, and it'll show you the appointments that she has.
That worked. Thank you.
Thank you. It's one of the challenging parts is that my screen doesn't look like your screen. So thank you for that. Let's see. Okay, I've got another question in the chat that says, how much should we be using the book and doing the readings? I expect you guys to do the readings, but keep in mind that I am not going to test you guys on things that are in sections of the book that I don't talk about. And so I want to keep the videos relatively short, because if I make them longer than 15 minutes, you guys get bored and stop watching them, which is pretty normal for anyone who's watching lecture videos and trying to take notes and think about stuff. And so because of that, the videos really focus on the big important pieces of things. Read the book to get the kind of little detail to make some connections between stuff. But if there's topics in the book that aren't at all talked about in my video lectures and that are nowhere in those learning objectives, you don't need to read them. And so I do expect you guys to look in the book. I don't expect you guys to read every single part of every single chapter that we're talking about. For certain chapters, I'll even list just the certain sections that we're going over. For other ones, you'll notice there'll be pieces of chapter sections that I don't seem to talk about in my video lectures. You don't need to worry about those. The ones that I talk about in lecture and the kind of like the structures that I talk about in lecture, make sure you're reading those sections of the book, because that'll help pull out a little bit more detail than I have time to cover in those short videos. Other questions?
I have a question. So are we supposed to meet tomorrow with you, with our team?
No.
Or
so
next
those
week?
case study appointments are completely optional. So I had originally thought about doing it where I made everyone synchronously meet with their case study groups and myself. The survey I had sent out about a week ago was about 50-50 split of people wanting synchronous versus asynchronous. And there was enough people in there that said they really didn't want to be meeting at the same time as everyone else because it was harder to schedule for them, that I didn't do that. So those are there in case your case study group wants to meet with me to talk about stuff, but you're not required to. And for this week, you guys don't even have your case study groups yet, so ignore that for right now. That's what I'm going to set up with the CATME that has to be finished by the end of the day today. So everybody will get their case study group assignments tomorrow, and then you guys can find time to meet to introduce yourselves and start working on the case study some next week. That's when you can decide if you want to meet with me at certain times. You're more than welcome to, but it's not required for anyone.
All right, thank you.
Any other questions? I've got one question up here about a problem with the play pause it videos. Johnny, I can talk to you on the side. I haven't heard of anyone else having any trouble with them. Has anyone else had any problems with the lecture videos? Got a couple here that Chrome worked, but not Safari. So yeah, I'll look into that for you, Johnny. I'll send you an email afterwards. Any other questions? All right, so yes, so you are required to record your case study group discussions. It's actually why I set things up in Teams. So once you have your case study group put together, I'm going to load everybody into their Teams group, and then you'll have a spot in the Teams page that you can all work together. So from that spot, you'll just say, meet now. It'll pull everyone up so you can video chat and talk with each other, but you'll want to record that. And so in Teams, when you highlight over kind of the bar that has your camera and your microphone and all that, you've got your little 3-dot options. There's a record button in there. And so you'll just record the conversations. That's mainly because I am giving you guys participation points. A big part of the case studies for me is getting you guys used to working in teams and talking about your ideas. Anyone going into health professionals will never be working in a bubble, so you have to get used to working with people. And so the key with doing it in Teams is the recording saves automatically to your team channel. So you don't need to worry about turning anything in or kind of trying to get a video uploaded to Canvas. I could just come in, play it quickly, check to make sure everyone seems to be there and participating, and give you guys your participation credit. So you should record when your case study groups have your case study discussion, just so that I can give you your participation credit. You guys are only required to meet with those case study groups for an hour during the week that your case studies are due, but you are more than welcome to meet as many times as you want to work both on case studies or the worksheets, or use it as a study group or anything else you might want to do. But I'm just looking for that 1 hour recording as your official group meeting. Other questions? Going once, going twice?
So just every single Thursday at 10, you'll be doing these lecture videos. They'll all be recorded, but it'll only be on Thursdays that you'll be doing this.
Yes. Yeah, so every Thursday at 10, I'm doing this. There's office hours times you can sign up to meet with me one-on-one on Wednesdays and Thursdays. And then there's case study times you can sign up on Fridays. And if those times don't work, if you guys need to meet with me, you can always just shoot me an email and we can set up a time to meet at any point. All right, anyone else? Okay, sounds like that's about it for the questions. I am going to stop the recording. So these recordings